Okay, so we are starting video number 4 for phylogenetic tree. And just remember these sequences here. And after that, our downloaded file is in our download. And if you remember this, how they turn around, like they end here, and then in the next side, it goes down and start all over again. So it doesn't go all the way to the right, okay? So let's see if we can find a way and make these ones really visible to us in straight line, in one straight line. All these 15 or 20 sequences, I'm saying 20 because we downloaded 20 and the other 5 they're missing, they may exist downward, okay? If we go further, drag down, some might end and some other might start because it depends where it matches. So it will then align them right at that particular location. But let's see if we can create a better alignment or visualize this file, this alignment into a different software where it can make it all linear, going from left to right all the way to the end of this particular file. So it will not be going downward like this, but it will be going from left to right. Okay, now it's going downward. When it finishes, then it comes turn around, go to the next line, and then keep going to the next line and the next lines. So this one block, all 15 reads here we see are going into the next block. So let's see the next step. Okay, so we are going into a website that I have already pulled it in. It's called the jalview.org. I hope it's visible and you're able to see it. If not, then you see it right there. Jalview, that's what you type, and .org. Okay, that's the main site. After that, you come to Jalview.js and you say launch Jalview.js. Okay, and you will see this console. And actually, we already clicked it again, so that kind of made it disappear. But you can say launch and see if it will get launched. I have this pop-up issues, so let's see again. And it's not launching. Let's do clear console, and there you go. So you have to clear that console. There were a lot of things written in there. All you have to do is click on that clear console, and now you have this pop-up thing right there. This kind of popped up, and all you have to do is go to File, and you say Input Alignment. Okay, Input Alignment, and you see from File. Okay, here you go. So now you have this created. So you need to now choose that file that we created earlier from multiple sequence alignment in the previous video. So all you have to do is say choose a file right there, and here you go. That's the one, MAFFT, right? That should be your file, starting with MAFFT. And you just click on that and you say open, and it should be, it's right there. The file is there, and then you have to say load. Okay, so let's see how long would it take. It's not a huge file, so it shouldn't take very long because there you go. So now what you see here is consensus, So it gave you your sequence, what it's supposed to be, the overall sequence that the program prefers to fetch from your alignment. In alignment, there are differences from each other as well. But if you look at these ones, see the TAAT, these are pretty similar. If you look at these, the box that I'm making around it is pretty similar sequence. Even here, some differences starting. So here, this T is different than this G, than this G. But this is what you need for phylogenetic tree analysis, okay? The rest you don't have to worry about it actually, what you see here. But this is the depth, okay? This is telling you how many reads are in this particular place and also giving you the percentage of the base. How many reads are calling C? And that is 33% is calling C, okay? Let me see if I can bring it a bit down. Okay, and if you keep going this way, so now you look at these reads, right? This one is one read, another two, whatever number. And if you then keep going to the right, it will keep going. See? So all these things are now in one straight line, and you can keep going to the right, and you see actually the occupancy. It means how much is occupied. Okay, this if you look at this, this is occupied quite well. These T's, right, or all these reads on this side. But if you look at this area right here, is not occupied. So if you look at the depth, it's giving you there is only one read here in this particular location. And for publication, this is quite good because you can create boxes around it and you can see or take snapshots even at this particular time and point to these locations where you are missing sequence or where you see events that they are of interest to you. So for me, see if you look at this, now this is of interest to me. This part is of interest to me. So I can show this in a box like this, and this one is now in a straight line alignment, and it keeps going. Some reads will end and new will start. So I believe there are 20 reads here, but not all in one place. So that's why the names are being missed, and you when you're counting downward, you don't see all of those reads. Okay, but now you know your cluster alignment, your multiple sequence alignment is there, and you can keep going to the right and it will end when it ends, okay? So the whole sequence is going from left to right, or you can say from right to left, but it is from left to right because the names are on the left side. Okay, so I hope this is clear, and obviously this step is not required for making a phylogenetic tree, but it is required in your life to visualize the multiple sequence alignment in a better form, in a nicer way. Here you can also see the numbers, the base numbers. Like if you know this is about 1620, the length of the whole sequence from starting from left up to here is 1619 up to this T, okay? Up to this C is 1620. That is base number 1620 starting from the left side. And it still keeps going. So let's see if we can go all the way to the right and see if we can end the sequence. There you go. So our total sequence now is about right there. This is the poly A tail of the mRNA since we downloaded the mRNA sequences, right? So if you look at it, there are about 1990, or you can simply say it's about 2000 bases from left to right. So now you have the whole thing and it's pretty nicely arranged, and that gives you power to make snapshots for your publications, for your thesis, or even to visualize the multiple sequence alignment before you make phylogenetic tree. So this is not a critical step in creating phylogenetic tree, but it is critical for you to see and visualize your data that you were working